100 days from today, we all will be writing the CSIR NET exam. Good evening, everybody. Please confirm in chat if you can hear me and see me too. Quickly, let me know. Okay, all right. Welcome to Biotechnica. Welcome to the webinar on strategies for 100 days of CSIR NET. Now, there are many YouTube pundits out there who are giving you strategy, and I'm happy you're getting a lot of strategies. But there's a problem with strategy. If there are too many strategies coming in, then it leads to analysis paralysis. And one person cannot give you strategies for all of them. Okay? You see, 13 units of CSIR NET, 13 different MSCs. Actually, truly speaking, zoology has a 360-degree topic approach from botany, cell biology, ecology. There are two different things, right? So what should be your strategy to qualify in these 100 days? That's something which we are going to talk about today. But let me ask you a question. If I give you 3 questions, okay? First question from part C, second question from part B, and third question from part A. Which one will you solve first and why? Tell me in the chat box. Everybody in the chat box, tell me, why would you solve which question? Part C, part B, or part A? Let me know in the chat section. Okay, all right. So what I see here, most of you are saying part C, some of you are saying part B, right? Now, let me be very, very clear. This is the first strategy which I'm going to tell you, and rest, of course, our subject matter experts are equally capable to share more strategies with you. But one very important strategy is, if you are unable to solve part C questions, you cannot qualify exam. So keep that in mind. 25 questions, 4 marks is equal to 100. That means out of 200 marks, part C has got a 50% weightage, right? So maximum time should be spent on solving part C questions. And that is the first strategy of the next 100 days. You don't need to solve PYQs. You need to solve only part C of PYQs. We'll save a lot of time there, right? Because if you're able to solve part C questions, automatically part B you can solve. There's no rocket science there. Now, the next thing here. At Biotechnica, we are coming up with the Raftar batch, where we are going to solve as many part C questions. We will go in speed, we will revise fast, and we will study only part C questions and of course concepts. We'll understand the concepts, solve more questions. More the number of questions which you are solving in part C, your chances of success is higher, right? Now I can see a lot of students who are there already from other batches also, Lakshya batch, Arjuna batch and Drona batch. So don't worry, for you people also we'll be coming up with the question modules. It is not that only for Raftar, but whoever has not enrolled, here is your last chance, because Raftar batch is starting from 11th of March. So without wasting much of your time, now I would like to invite our, not just 1, 2, 3, we will have 6 speakers today who will talk in elaborate manner on every portion, every strategy of the syllabus, which you should be focusing on in the next 100 days. But on the top of it, if you attend this webinar till the end, then we are going to give away a list of all the top 100 topics which you must focus in this exam in the next 100 days. So 100 days, 100 topics makes sense, right? So we are going to give you a free ebook PDF, which will have the 100 topics which you should be studying in the next 100 days, right? So do not forget to attend this webinar till the end, where we will give you this particular free download. All right. So now all the students out there who are preparing for CSIR NET, you have to keep this in mind that this exam is not here to test only your knowledge. It is here to test your strategy. It is here to test your time management skills. It is actually looking for a 360-degree researcher. They're not looking for a person who has only concepts. They're looking for a person who can apply concepts. So listen to this webinar till the end, and I'm sure you'll have 100 plus actionable insights, and that will empower your next 100 days. So all the best, and I will see you at the end. Thank you.
Hello, everyone. Good evening. Good evening, everybody. Good to see you all here. So, today we are going to have a 100-day preparation masterclass for CSIR NET Life Science. And my portion, next 10 minutes, I'm going to talk about setting the foundation for the exam. Okay, so in that foundation, what all things I'll be talking about? I'll be briefly explaining you the exam pattern, and I will be helping out with the syllabus, not in detail, because our next faculty will talk to you in detail about the syllabus and the important topics. And next, I will tell you about the reference books. Okay, so let's get started. Now, for those of you who are not aware of the exam pattern, so this in CSIR UGC NET is a single paper MCQ based test, which is CBT, that is computer based test conducted by NTA. The duration of the exam is 3 hours. There will be total 145 questions divided into 3 sections, A, B, and C. And total marks that you can obtain, the maximum marks that you can obtain for the paper is 200. Now, what about the marks distribution? So, total number of questions are 145. This 145 question, as I just told you, is divided into part A, B, and C. Part A comprises of 20 questions, but you don't have to take all the 20. Okay, you have to attempt any 15. Now, in case you are attempting 16 or 17 questions, don't think that you will get extra marks. First 15 will be scored. Okay, that's very clearly mentioned in the brochure. So part A will have 20 questions, out of which you have to attempt only 15. Part B will have 50 questions, out of which you have to attempt only 35. And part C, which is the having the maximum bouquet of questions, it has 75 questions, and you have to attempt only 25 out of it. Now, what about the marks distribution? So part A and part B gets plus 2 marks for every correct question and minus 0.5 for every wrong answer. And for part C, every question can fetch you plus 4 marks, but every wrong answer can also give you minus 1 mark. Okay, so this is how the questions are distributed. Now you have to be very careful when you are attempting the exam that you don't go beyond these numbers. Okay, because otherwise you will end up wasting this number, wasting your time, and these extra questions that you might be attempting will not fetch you any marks as well. The paper breakup. So part A has general aptitude questions. Okay, so general aptitude, you don't have to go for the complex mathematics part. The 10th level mathematics, okay, whether you are talking about speed and distance type of questions, relative speed kind of questions, graphical analysis, series formation, puzzle, geometric questions. Okay, so those are the questions which are asked in part A. Part B is subject based, that is based on the CSIR NET Life Science syllabus, and it is fact based. Okay, direct fact based questions will be there. And part C is the one where it is analytical and conceptual question and experiment based. Most of the question you will see that they will give you an experimental data. So the main thing that they want to check in part C type of question is how good are the aspirants in taking or analyzing the results. Okay, because anyways that is what you are applying for, right? You want to go for research. So the experiment that you are doing, the result that you are getting, how good are you in analyzing that? That is the skill that they want to check in part C. Okay, now once we have understood the paper breakup, so next comes syllabus. Okay, now I'm not going to tell you the detail about the syllabus here. I will request our team to post the link for the syllabus. You can see the link in the chat in the chat box. Okay, so yes, here is the link which has been posted here. So if you are not yet aware of the syllabus, please click on the link. 10-page syllabus is available for download. You can download it, keep it with you, and go through it seriously so that you can prepare better. Okay, now once you have got the syllabus, now what should be your next? So this is showing the cutoff for December. Okay, so as you can see, for the unreserved category, the cutoff for JRF was 99.21 percentile. For lecturership, it was 98.05 percentile. Now, why am I showing you the cutoff for December? Because this is your target. Okay, your target is not only this number, it's to score beyond that number. Okay, that make it very, very clear, because when you are starting your preparation now, your target should be absolutely clear to you. So this is what is your target. Your target will be to cross this number, because mind you, every session the cutoff percentage keeps on going high. Okay, so you have to score up you have to get a score which will have higher percentile. Okay, for sure, if you want to clear the June CSIR exam. Okay, now you have your syllabus, you have your the exam pattern, you have understood, you have set up your target as well. Okay, now we need to get ready for the war. Okay, how to get ready? By achieving, or by getting more and more information, more and more knowledge. Okay, now who can be best in your preparation? The reference books, right? Now, how to choose a reference book? Because you see, the CSI syllabus is divided into 13 units. Each unit can have multiple standard reference books are there for each and every unit. Okay, so how to choose the reference book? So I'm going to give you some tips and tricks for that. Okay, so first of all, focus on the CSIR NET Life Science syllabus. Okay, go through the syllabus, see what all units, what all topics are included, and then check for a reference book which combines or which has all of those topics. Next, content clarity and organization. So look for the books where all the contents are given in a language that is easier for you to understand, and also see how the information are arranged. Okay, whether they are arranged in a flow like manner. Okay, so that when you are reading it, you can understand the whole picture. Okay. Next is recency of the information. So when you have the same book and multiple different editions, always go for the most recent edition. Okay, why so? Because when you are choosing most recent edition, most of these books will include the recent research or the recent findings which are going on in that field. And mind you, CSIR question paper do they are based on this recent information. Okay, so those books will be able to give you the better and recent informations. Then look for the books which will give you additional features. Additional features like what? Mock test, questions at the back of each of the chapter that will help you prepare. Or nowadays, a lot of books give you the animation series as well. Okay, so what all animation series is the how the collection is given? Sometimes they give you the QR code, and with that you can get the access to their animation. So this extra feature is also something which you should consider when looking for a reference book. And definitely, if you're still confused, you can look for recommendations. You can ask your seniors, you can ask your fellow aspirants, also you can ask your teachers. And then finally, compare and choose the best reference book that works for you. Okay, now mind you that it's not required that everybody has the similar choice. So whatever works for you, it might not work for other. Okay, so you have to take that decision and choose the book which is right for you. All right. Now, I would request our team to post the link of the video which has the detail about all the reference books. Okay, we already have a video in YouTube where the detailed information or multiple books for each and every unit is mentioned there. The link is already posted, so you guys can go through it and you can get a help on which all reference books are there for the different units. Okay, now once you have got that, so I would like to tell you about how to use this reference books more efficiently. Okay, so I'm going to divide it into 3 parts. So it will be before you start reading, next while you are reading, and last will be after you are done reading from the book. Okay, so what all things you should do to get maximum information from, or maximum useful information from these reference books. So before you start, so don't just start reading a book from page 1 to page last. That will be a complete waste of time. Consult the index and table of contents, match it with the syllabus that you have, and then go to the go to those exact part of the books where the content is there. Okay, content for the topic that you are looking for is there. That will save a lot of your time. Now when you are in that topic also, you don't have to study each and everything mentioned there. Okay, so you can skim and scan. How you can do it? By focusing on the highlighted words, by focusing on the larger font words, by focusing on the on the highlight, the headings. Okay, now along with that also, nowadays, like if you're looking for the newest edition books, so always read the boxed information. Okay, the information which is given in box. So when we are in universities, we kind of skip those boxes, right? We feel that those are not relevant information. But for your CSIR net exam, I tell you that this boxed information are the one which are the most recent researches or the findings that are mentioned there. Okay, so always read those boxed information that is given in the reference book. Now while reading the book, don't just read the words, actively engage with the material, understand it, and understand it and try to link it with the other concepts that you know. Okay, because ultimately you are reading biology, you are studying life science. Okay, everything is linked. So try to link the topic so that you get the better view of the larger picture. Pay attention to diagrams, figures, and tables. Okay, these are very, very important. Sometime in exam, exact diagram, exact table is given. Okay, they just omit out some labeling and you have to find out, you have to label that. Okay, those kind of questions have come. So do pay attention to this diagram, figures, and tables. And this diagram, figures, and tables, they also give you information in a very compiled way. Okay, so it is easy to grasp. All right, so always pay attention on these things and also use these things for your revision. But after you are done, okay, now what you have to do, you have to summarize and review. So how you can do it? You can do it by writing notes, or the best way will be if you have made a peer group of studying together, so you can teach each other. Okay, every day take turn and teach each other, because when you are teaching, that time you are converting the bookish information into your own language according to your own understanding and explaining. Okay, that is a very good way of uh, keeping the information or memorizing thing or embedding it in your memory. Okay, and next thing, the most important thing will be practicing questions in your daily timetable. Now from 100 next 100 days, keep out time at least 2 hours every day for practicing questions. Whatever topic you are studying today, whatever topic you studied yesterday or last week, every day you should keep 2 hours, minimum 2 hours of practicing questions. So with this, guys, get to work, keep working, and do not stop for at least next 100 days. And with that, I'm going to pass on control to our next experts to explain you about the important topics. Thank you, everyone.
So, hello everyone. Am I audible to all of you? I hope so. My screen is also visible to everyone. Right. So, thank you, Tithi ma'am, for giving the a broader idea about the CSIR net pattern and the syllabus and the mark distribution, reference book, everything. So this session is all about the important topics from each unit. Okay. So to begin with, we have the very first unit, that is biochemistry. So biochemistry is considered to be one of the important unit from CSIR point of view. Right. So already the syllabus, you are aware of this. Now I'm just going to give you the important topics. So from biochemistry, remember the structure of proteins, nucleic acids, amino acids are important. Okay. Interruption. So yeah, I was just speaking regarding the important topics from each of this unit. So to begin with, we have the biochemistry part, right? So in that case, the important topics includes your proteins, the structure of proteins, nucleic acids, and even amino acid structures are also important. Apart from that, stabilizing interactions are there. Then, when we speak regarding the biochemistry, obviously numericals plays a very important role, right? So numericals basically comes from pH, buffer, reaction kinetics, thermodynamics, and all. Then, other than that, we have metabolic pathways that plays a huge weightage when it comes to CSIR net exam. So, remaining important topics, you will get a link. All you need to do is click onto that link and download those topics. So, all total, you'll be having 100 topics you'll be getting from there. Okay. Then, now the thing is that how you are going to prepare for this unit 1. Right. So here are some tips and tricks that you can focus on. The very first thing is that be clear with the concepts of atoms, molecules, bonds, functional groups, oxidation reduction processes, nucleophiles, and stereochemistry. So basically, when we say biochemistry, it is the base which onto which other units are constructed, or we can say connected to each other. So you have to be very clear with the basic concept. Next, when we look into the metabolic pathways, either you can go for manually, you can write the metabolic pathways, or you can take images from standard reference book. In fact, when you are going for this images, you can highlight the important enzymes, the location with different colors. So when we are using different colors, obviously we will attract and the things will be retained in our mind for longer duration. Then, when it comes to comparison tables, yes, you can go for comparison table for ABZ forms of DNA and different types of enzyme inhibition. As I mentioned about numericals, practice questions regarding molarity, normality, and about unit conversions. Then graphical based questions also come from unit 1, that is regarding the Michaelis-Menten equation, the Line-Weaver-Burke plot. So for that you can use standard reference books. I hope so you are aware of the standard reference book, right? So which is the best book for biochemistry? You have heard in the first session, right? Yes, we have Leninger. So Leninger you can focus on and you will get the important graphs from there. So here this is just a sample for you to show that a comparison of different forms of DNA. So if you make this comparison table, you will be able to understand and retain the things within a shorter period of time. Like for last moment preparation and all, this is going to help you out. Apart from that, if you look for the metabolic pathways, you have glycolysis, gluconeogenesis, starts from glucose. Of course, the two processes are not same with minor variations in the enzymes. Right. So when you will go into details, you'll come to know the differences. But maintaining this kind of chart will be easy for you. And from here, even you can extend from pyruvate, you can enter into the TCA cycle, then you can go for ETC. So you can extend accordingly. So if you make a big chart of this, that itself will be more than enough to remember the things. Moving on to the next unit, we have cellular organization part, that is the cell biology part. So undoubtedly, protein targeting plays a very important role. So by hook or by crook, you will get one or two questions from this part. Apart from that, structure of chromosomes, chromatin, cell cycle regulation are other important topics. Now let's check it out how we are going to prepare this cell biology part. Here, in majority of cases, specifically if I say the protein transport part and all, better to use diagrams. In certain cases, you can use flowcharts as well, but diagrams is recommended. Okay. Then, when it comes to cytoskeletal proteins, inhibitors, motor proteins related to that, you can use comparison table. For genome organization, again, diagrams are recommended because when you make diagrams, you will understand the different orders of packaging, right? The first order, second order, third order and all. So that will be more clear when you go for diagrammatic representation. Then when it comes to cell division, that is related to cell cycle, basically this particular topic is combined with techniques, right? This is combined with techniques. Other than that, some other topics will be connected. For example, we have apoptosis, cancer. So nowadays the questions do come in the combined form. So, for that case, you have to be very clear with the basic things. What is a cell cycle? What are the different proteins involved? And the corresponding functions. Because the mutation of those proteins is something that is related to cell cycle. So, this becomes a very important part. Then numericals, compared to biochemistry, here numericals are least expected. But yes, still, if it comes, maybe it will be related to growth curve, bacterial growth curve related questions you can get, and that you can easily get it from Prescott. Okay, so when it comes to numericals, the only way out over here is practice. Keep on practicing the numericals. Moving on to the third unit, before to that, just showing you a sample. See, this is an image of protein targeting into the nucleus. So by looking into the image, I guess you are able to understand how the proteins are getting targeted, how they are tethered to the cytoplasmic filaments, how they are entering into the nucleus. Similarly, this is related to lysosomal targeting part. So lysosomal targeting, here also you can see how the proteins are crossing the Golgi apparatus. With the help of some vesicles, they are entering into the endosomal compartment. So that's the reason I told you, when you go for protein transport, it is better to go with diagrams. Next, moving on to the third unit, that is fundamental process. Now, this is one such unit in which all the topics are equally important. So, whether you take into consideration replication, transcription, translation, or the regulation of gene expression, all the four parts are equally important. Now, how we are going to focus on this particular unit? Here, you can go for visual aids. That means the best way to understand these process is animation. So animations will give you a better input regarding different processes, how the enzymes are coming, how the DNA is unwinding, how the DNA getting double stranded, how the transcription factors are getting recruited, how they are bind and which portion of the DNA they are getting bind. Everything will be clearly mentioned through animations. Other than that, here also you can focus on diagrams which you can take it from the standard reference books. After that, the second way is chunking down. That means you have to break down each process into smaller and more manageable chunks. That means whether you take into consideration replication, or you take into consideration transcription, or you take into consideration translation. So in all the cases, we have certain phases, like we have initiation, elongation, termination. So what you can do, you can separate the parts. Remember the individual factors that are involved in each phase of that particular replication or transcription or translation, and try to remember. Then you can compare and learn all the processes. Compare and learn all the process means, as we know that this central dogma of life is common when it comes to prokaryotes and eukaryotes. Of course, there will be minor variations will be there in terms of enzymes or the transcription factors or the factors that are involved. Otherwise, the overall process remains the same. So for that purpose, what you can do, you can compare and learn. So if you look over here, see, this is the replication in prokaryotes and eukaryotes. Overall, the process remains the same, but what is different? The difference is in terms of enzymes. Similarly, take the case of transcription. So in case of transcription, prokaryotes, nothing much is there. But yes, when it comes to eukaryotes, you'll find a lot of transcription factors are involved. When it comes to translation, yes, again, you can compare the bacterial part and the eukaryotic part. So basically, when it comes to these kind of processes, better go for comparison. Apart from that, you can go for diagrams, like for example, lac operon, if you see, where the gene expression is higher, where the gene expression is lower, and in which case the gene expression you are not able to visualize. So just by looking into the image, you are able to identify what is the difference. So that's why I had mentioned diagrammatic representation also helps when it comes to unit 3. Then moving ahead, we have the fourth unit, that is cell communication and cell signaling. From this unit, you can ignore this part or you can combine this with immunology part, because more or less the diseases are quite relevant to each other. So that's why you can combine these two things, and other subunits, whether it is cell signaling, cellular communication or cancer, all are equally important. Yes, immunology as well, because when it comes to immunology you have to cover the entire thing, because if you don't understand the basics, it becomes difficult for you to understand further processes. So you have to cover all the topics. So how you are going to focus on? So when it comes to signaling, more or less in any signaling pathway you go for, you will have ligands, you will have effector proteins. They have their own set of second messengers and protein kinases. So depending on that, different names will be given. Like when you go for GPCR pathway, you have 4 classifications of GPCR pathway. So, what is the difference among the pathways? These are the components that will be varied. Then, when it comes to cellular communication part, here basically you will be studying about how a cell to cell interaction takes place and how a cell to matrix interaction takes place. So, in that case, how this interaction takes place is via different forms of junctions, different forms of cytoskeletal proteins. Then some intermediate filaments, actin filaments will be involved. Other than that, adapter proteins will be there. So combining this, you can create a table. In fact, tables are also available in the reference books like Lodish, Albert and all. You can also have a check on to that. Then finally, when it comes to apoptosis and cancer, focus on diagrams as well as flowcharts. So how you can prepare for cell signaling? See, this is the 3 signal transduction pathways. As you can see, this is your JAK-STAT pathway, this is your GPCR pathway, and this is your receptor enzyme pathway, that is your insulin signaling. So by looking into the image, you are able to understand, okay, we have got a ligand that goes and binds to the receptor, autophosphorylation takes place, transcription factors are activated, so NLS will be exposed and enters inside the nucleus. Similarly, here also by looking into the pathway, you are able to understand that yes, this is how the signaling takes place. So better whenever you study the cell signaling part, always go for colorful diagrams. Other ways, if you want to note it down or if you want to do it by your own, you can make it in the form of a flow chart, right? So this is one example of that. So this is an example of a GT pathway, that is the rhodopsin pathway. You can create flow charts in this way. In fact, here you can highlight few things like hypopolarization, hyperpolarization, what is the enzyme that is responsible for the breakdown of CGMP, and finally how you are relating to vision. So few things you can highlight when you go for these kind of manual pathways. Then coming to apoptosis, yes, apoptosis related questions do come, but not on a regular basis, but yes, it comes on every alternative times. So if you compare, so basically here comparison is carried out, compare and learn. So how you can compare and learn, as you can see, 3 different pathways are there, but everywhere it ends with caspase 3 and downstream remains the same. So all you need to do is remember the first part. That means the initial part. Like here you are having the death ligand and receptor. Here there is some mitochondrial changes occurring. Here some serine proteases are released. And the major difference among them will be the initiator caspases. Okay, but the executional caspase will remain the same. The other 3 is the initiator caspases. Okay, so like that you can compare and study when it comes to apoptosis. Then questions related to nematode and vertebrates do come. So when it comes to apoptosis, certain proteins are involved, like regulatory proteins are there, adapter proteins are there, and effector proteins are there. So the names are different, but the function remains the same. So again, this will help in remembering that part of your exam. Then apoptosis, as you might be aware that it is made up of BCL2 family of proteins, right? So BCL2 family again classified as pro-survival and pro-apoptotic. So you can use some code words or mnemonics in order to remember and differentiate the names of these proteins. Cancer, again, another important topic. Basically, the questions are experimental questions are coming from cancer, which is again connected with techniques or some other topics related questions do come over here. So remember here, different genes are involved. So basically, cancer happens because of unproliferation, like uncontrolled proliferation of genes, right? So what are the genes that undergoes mutation? What kind of mutation it is, and what are the properties of the mutant gene. So everything is accommodated in this single table. This will help you in remembering the cancer part. Then nowadays the questions very frequently have seen with respect to Pasteur effect and Warburg effect. Right. So by looking into this image, you can easily differentiate this is a normal cell and this is a cancer cell. How? Because you see that maximum of the pyruvate is utilized in oxidative phosphorylation. So that is what happen on regular basis. But when it comes to cancer cell, what happens? Majority of portion is provided to lactate fermentation. Right. So just by looking into the image, just by understanding this, you'll be able to get a 4 marks easily, because questions, even though they are asking in 4 marks, but those questions are direct questions. You can easily fetch those marks from here. Next is the fifth unit, that is your developmental biology part. Here also there is no such topic which can be skipped. Of course, programmed cell death can be covered under unit 4, so you don't have to focus it over here. And again, aging and senescence, when it comes, again it is a part of unit 6, plant hormones are there. So some part of it will also be discussed under there. So you have to don't put much effort on this. But yes, the basic concepts of development, gametogenesis, morphogenesis in plants and animals all are equally important, which you can never skip it. So what are the ways you can remember it? Here also you can follow the same pattern, that is comparative charts. That is when it comes to stem cells, the types of specifications, the fate of germ layers, types of X, what is their cleavage pattern. For everything you can create a table form. Then other than that, the question majority of times which you get in the form of 4 marks is mutation related. So mutation related to C. elegans, Drosophila, amphibian, and tetrapod. The only way which I can say you can fetch marks from here is memorize. And questions come directly from Gilbert book. You don't have to look for any other sources. When you compare with other units, whether it is your biochemistry or cell biology, you have to go for number of reference books. But this is one such unit, one of the unit where you can focus only on Gilbert book and easily fetch marks out of that. Graphical based questions also do come from this unit 5, developmental biology. This is just an image of a comparison between gastrulation that occurs in the chick embryo to that of an amphibians. So the image comparison will help you to remember the things better. Then even under developmental biology also, you will come across the signal transduction pathway, what happens when Wnt signaling is not there and what happens when the Wnt signaling is there. So what are the enzymes? What are the proteins that are involved in the cascade? So that will help you to differentiate and remember. So not only Venn pathway, hedgehog pathway is also there, delta notch pathway is also available. So these are basically, these are the three important pathways which comes under the developmental biology part. Next, what we are having, ABC flowering. Yes, the plant developmental biology part, the questions do come from here, that is ABC flowering. So we are aware that plants, the flowers are made up of four holes, which will be corresponding to different structures. And that structures will be coded by specific genes. So basically, again, mutation based questions do come from here. So they may ask if mutation occurs in A, what happens, or AB combination or BC combination, or what happens if all the three genes are mutated. So what will be the structure of the flower? So those kind of questions do come from this part. Next, we are having Unit 6. Well, Unit 6 is a unit. Sometimes people will prefer to choose either Unit 6 or Unit 7. Either of this is a choice depending upon your background, depending upon your interest. So when it comes to Unit 6, you have these many subunits, and what subunit which you can skip is nitrogen metabolism part as well as the stress physiology part. Okay, so these are the two parts. I will not say completely skip because questions do come from here, but usually comes in the rotational basis. Rotational basis means not always, but sometimes you can get questions. And how exactly you are going to prepare this particular subunit? Well, when it comes to photosynthesis, respiration, stress physiology part, you can go for mind maps. You can also go for memory play cards to retain the concepts through revision. And when it comes to plant hormones and secondary metabolites, you can always go for comparison of different hormones and different types of metabolites that are involved. Then this is again an important part, C2, C3, C4 and CAM pathways. Here also what you can do, you can compare and learn. So I will just show you one example. See, this is your CAM pathway. So here, what happens in the dark condition, what happens in the light condition. So during the dark, as you can see, stomata is open. So easily the carbon dioxide comes inside. But during the light, what happens? Stomata is closed. So that is being restricted. So just by looking into the image, you will be able to identify what is the difference. So these are the tools or these are the techniques which you can use for the last moment or last minute preparation. Other than that, as I told you about the hormones, you have this table. So along with that, you can add extra columns regarding the signaling part. You can add, and also you can add the biosynthetic part. Then you can also add the inhibitors, the synthetic forms, natural forms. Those columns also you can add. You can create a table which you can compare and study. Okay, auxin is the tryptophan is the precursor of auxin. Cytokinin, gibberellin, the precursor is different. So if you add extra columns into this table, again, it will be easy for you to compare and study. Then secondary metabolites. Secondary metabolites, what is the problem is that usually we get confused with which compounds comes under which category. So this kind of mind maps or flow charts will be helpful for you to remember. So what all things comes under terpenes, what all things comes under phenolic compounds, what all things comes under your alkaloids, whether it is your nitrogen containing compound or it is your sulfur containing compound. So overall speaking, when I say unit 1 to 6, all the units are important, and the common techniques that you can use over here will be the tables, the diagrams and the flowcharts. So that's it from my side. Now I am passing the controls to Somrhita ma'am. She will be continuing with the remaining subunits.
Good evening, everyone. Thank you, Divya ma'am, for that wonderful insight. And I'm sure the students by now, they are getting a very, very clear idea about how to prepare with the first six topics and what we mean by the flow chart, what we mean by studying by the diagrams, what we mean by, comparative tables, right? Aren't you getting an idea? So basically, I will continue that strategy from unit 7 onwards. But before I get into the units, I want to give you a very important idea about how to prepare your CSIR net. What are the steps you should follow while you are preparing? And this Bloom's taxonomy is actually going to help you. This is very, very effective. And this is something that you should apply when you are studying. Okay, because just studying will not give you the desired marks. So first step in this part is definitely to remember. That means here you have to refer to the books. Okay, the reference books list you have got already, you have got the PDF. So whichever topic you are studying, get hold of that particular PDF, or if you have the hard copy, whatever it is. Okay, and read from that book which are the important topics, select that. Okay, and read from there. Look at the diagrams, look at the tables, look at the boxes which are there. Okay, and once you have read, before you just close the book for the day, try to recollect what you have studied. Okay, before you just close the book and go over to the next topic, don't do it like that way. Recollect. Okay, because one thing is, no matter which unit you are preparing, whether there is more numericals from that unit, whether there are more conceptual questions from that unit, you have to remember the concepts, right? That is very, very important. So the remembering part is definitely applicable for all the units. Okay, so recall the facts and basic concepts while you are studying, and that study should be a very conscious study. Okay, not just reading the lines. Okay, now once you have studied it, you have just recollected, okay, what I have studied, and what are the important points. So what you can do is, either you can just recollect, or you have a rough paper and just scribble the points, just scribble it down. Okay, that way also you can do it. But that moment revision is definitely important. It is going to help you to remember. Okay, after that, try to explain the ideas. It might not be just immediately after you have studied. Okay, you can do it if you have time. You can definitely go for this explanation. Like when I told you, before you close the book, recollect, right? So that recollection can be a way of explanation also to someone. Now remember, no matter how difficult the topic is, if you can explain that topic to a class 6 student, okay, who doesn't have any idea about it, okay, if you can explain that topic, if you can make that person understand, that means you have a very, very clear concept about that topic. In fact, when you are explaining, many difficult concepts becomes easy. Okay, and that helps you to remember again, that you will never ever forget when you're explaining. Not only that, when you're explaining, it's a mode of recollection also. So whenever you're stuck, you can definitely go back to the book, or the note you have made, or the flow chart, whatever the tools you're using for studying, you can definitely go back, have a look. Okay, so explain. Now it's obviously, I'm telling you that you have to explain to a class 6 student. Now that just means that you have to explain. Okay, so self explanation. Okay, so explain it. Again, you can scribble while you're explaining, like how we teachers always do, right? We write okay, while we are explaining. No matter how, even if we are using a presentation, PowerPoint presentation, we always write, we scribble something, okay. So that way, that will help you to understand the topic very well, and this is very very important when you are preparing for any competitive question exam, particularly CSIR, okay, where more than memorization, analysis is important. And Shekhar Sir, in the start of the webinar, he has already told you that part C is important and why it is important, right? Okay, so when you are preparing for part C, understanding is important. Next is apply. So what you have understood, you apply it. Okay, now how will you apply? Go for the previous year question paper? No. Application doesn't straight away means go for the MCQ, go for the previous year question paper. Definitely you will go. But when it comes to application, any kind of question, and I always recommend, go to the back end questions of every chapter. Whether there are any MCQs given or whether there are statement based questions, go through those questions. Even if it is a statement based question, try to answer it, because it will help you to understand, it will clarify your concept, it is a self uh, you know uh, self judgment that you're making, okay, how well you have prepared, how well you have understood. Okay, so always try to apply that, use that information. All right, many informations are connected, many informations are linked, one chapter information is connected to another chapter information. Try to connect that. Fine. And then comes to analyze. All right, analyzing is, you draw connections among ideas. Like for example, I will tell you, there is a connection between unit 2, unit 3, unit 8. Like I'll just tell you, like in unit 8 you have a chapter as mutation. Okay, now mutation is in unit 8. You can connect, when you're studying mutation, go through repair, DNA repair. Okay, go through DNA repair, which is under unit 3. When you're studying DNA repair, go through the cell cycle checkpoints. Okay, when you are studying the cell cycle checkpoints, now this is under unit 2. When you're studying the cell cycle checkpoints, this will lead you to apoptosis as well. Okay, now apoptosis, this is connected to your unit 4. So see, just picking up one topic can help you revise so many connected topics. So analyze the thing properly. Because if you notice in your part C questions, many a times you get the concept from more than two topics. They have, clubbed the two concepts together. The concepts are related to each other. They have clubbed it together and they have presented that to you. Okay, when you are studying immunology, definitely study ELISA technique. Okay, antibody, ELISA technique, that is connected. So find out these connection points, all right, and analyze. And of course, after that, you have to evaluate. Now, when you are evaluating, this is something when you have to take a decision, okay? You have to argue, you have to defend, you have to judge. And how you can do it? This is the time you go for the previous year question papers. Okay, all the previous year's question paper, this is the time when you take it up. Okay, because when you're solving the previous year question paper, you are not just judging the concepts. Okay, you're not analyzing the concepts, you are not self judging. Okay, like what is your standpoint, how well you are prepared or not. You are also practicing time management, okay, which is very very important. So till your CSIR exam, these are the way, okay, that you should follow when you are preparing yourself.
Now, I'm sure CSIR is just not your target. That is the fundamental target. That's a first target you have. But after that, everyone is aspiring to do some PhD work, right? And that is why you are giving the CSIR exam, right? Now, till your CSIR, it is about evaluation. But what after the CSIR? Or not after, I will be wrong in saying after the CSIR. Basically, once you have qualified CSIR and you're approaching a particular lab, okay, sometimes the guide tells you to write an SOP or give some project idea. Okay, so that means whatever you have studied so far, you have to create something out of it in order to apply in a particular lab. Okay, so while you are studying, remember, you are just not preparing for the CSIR, but you are also preparing for the ultimate destination. Okay, so your preparation should be of that standard, and that is why understanding concept, applying the concept, okay, linking the concepts is very important, because when you're ultimately designing a experiment, okay, and these kinds of question again you will get to see in your part C. Many a times they give you experimental question, they give you a situation where they say that a researcher is working on so and so, and this is the conclusion he has drawn. So, what can you conclude out of it? Okay, that kind of question is also given in your CSIR exam. Okay, so you have to connect the concept, because ultimately that will help you to create a project idea. Now, having said that, let me get into the units. So till unit 6, Divya ma'am has already discussed. So I will, in very brief, I will discuss about unit 7, 8, 9, 10, 11, and 12. Now here again, time management is very important. Remember, you have 100 days, right? And you have 100 topics to prepare at least, right? Now, where you should give emphasis, where you should not give emphasis, how much time you should allot to a particular subject, how much not. This is something, this is a strategy which you need to prepare. Okay. So, for that, always remember that there are some topics which are very, very important. At least in this section, unit 8 is very, very important. Okay. This is a topic that is going to bring marks to you. Unit 10, Unit 11, and definitely Unit 13. All right. Now, when you are taking the first 6 topics, Unit 1, 2, 3, 4, 5 is very important. 6 and 7, you can make a choice. Right? 6 is talking about the plant physiology, 7 is about the system physiology. Now, those who are strong in animal physiology, those who have already studied some animal physiology during your BSc or MSc, definitely go for Unit 7. Okay. Those who have some knowledge about plant, you can definitely go for Unit 6. Now, those who don't have any knowledge about any of this, you will make a choice. Okay, where you're comfortable in, make a choice, 6 or 7. Right? Now, apart from that, all these units are very, very important. Okay. Now, Unit 1 is also very important, but Unit 1 is quite large. So, if you don't get time to prepare all of this, then you can definitely make a choice between Unit 1 and Unit 13, because Unit 13 itself is very large. Okay. Otherwise, majority of the units, all these units are very important from CSR point of view. And in the second set, Unit 8, 10, 11, and 13 are important. Okay. And as I told you, 7, you can make a choice. 9, only some selective topics you can do. Okay, don't have to go for all the topics. Some selective topics you can do. And if you stay till the end of this webinar, obviously you're going to get the list of the 100 important topics, where we have already mentioned the important topics from your Unit 9. Okay, what I mean, not only Unit 9, all the units, but definitely you will get it from for unit 9 as well. Applied biology also only few, just 2-3 topics you can study from applied biology. Rest of it you can skip. Now, this is a part, these two units. These are two units who are very important. Both the units are important, and both the units are very easy. Very easy. Okay, unlike inheritance biology, which is talking about genetics, unlike unit 3, which is cell biology, unit 4 talking about signaling and all, 10 and 11 is very easy. So definitely when you are preparing for these heavier units, okay, 2, 3, 4, 5, 8, 13, 1, unit 1, okay, along with that keep 10 and 11, but you can allot lesser time for this because it will not take much time to understand the topic. Okay, it will not take much time to understand these topics. In fact, when you are very tired of studying all the these heavy weighted topics where a lot of concept, lot of understanding is required, you can definitely as a break, okay, you can take up ecology. Take up the book, read it like a storybook. That will also do. That will also do because you are going through the concept, and trust me, it's so easy that you will be able to understand. Okay, same with your evolution as well. Now, what can be your study technique when you're taking up these units? So obviously the standard technique, the very first should be the self-explanation for all the units, Okay, self-explanation is a must to analyze yourself. Apart from that, definitely you can go by the diagrammatic analysis, diagrammatic study. You can go by the numerical study, flow chart, compare and learn point, and the mnemonics as well. Like for example, unit 7, when you're studying about the animal physiology, you will come across a lot of diagrams which are self-explanatory in its own way. Okay, if you just look at the diagrams and the ligands below, you will automatically revise 50% of the concept. Unit 9 also you can do it by the diagrams. Unit 10 as well. Okay, unit 12 go by the flowchart. Like for example, you have the plant tissue culture. Okay, you have the plant tissue culture or animal tissue culture. There are a lot of techniques which are discussed. Okay, so when you're studying the techniques, whether it the callus culture, whether it is the cell culture, okay, whether it is the protoplast culture, just use a flowchart to memorize the technique. It's as simple as that. Okay, it's very simple. Numericals, you get a lot of numericals from unit 8, few numericals from 10, and few numericals from 11, but 8 is all about numericals. And many students say that genetics numericals are very difficult, but my dear students, I want to tell you that if you have a clear concept about genetics, okay, right from the fundamental to the advanced part, the numericals are not at all difficult to answer. They are very simple numericals. Okay, but you have to have the concept clear. That is a trick to solve the numericals. One and only trick. From unit 10 you can get few numericals, like from the you have the population growth. Okay, population growth. From there you can get numericals. You can get numerical from your community structure, like when you are comparing or when you are studying the diversity. Okay, diversity of a community. This is a place where you get numericals. You get numericals from your variation, mean and variation also. Okay, life table, which again comes under your population chapter itself. So very few numericals, and they are very simple again. Unit 11, there is one section where you need to study the numericals, and that is your population genetics. Okay, under this section only you have the numericals, nowhere else. The rest of it is all about concept. Okay. Otherwise, just concept and some experiments. Okay. This is the only place where you get the numericals. Now, when you're studying for unit 9 and 13, it's always better to go for the comparative study. Okay, always better to go for the comparative study, because there are like in 13 you are studying many techniques, isn't it? You are studying very many techniques, like you are studying microscopy and all. Okay, you are studying spectroscopy, you are studying immunological techniques. So it is always better that once you have gone through those techniques, you compare the techniques and see what is the principle, what is the principle of that technique, what is the working principle, what is the outcome, what kind of compounds can be screened through that. Okay, these kind of comparative study is a must for unit 13. For unit 10, since it is mainly concept based, you can definitely go for the point studies. Like, so when you're studying, actually make some points, okay, bullets. Just don't write sentence after sentence, just the important words, mention the important words. Okay, for unit 11 also the same thing. Now in unit 11 I will show you also for your paleontological era. Okay, you can use the mnemonics to memorize the eras, the periods, the epochs and all. Okay, so basically these are the study techniques which you can apply for the different units here. And always remember one thing, when you are studying, no, the very uh, effective way of studying is to follow a 987 rule. Do you know about the 987 rule? It says that you study for 9 hours, okay, you sleep for the 8 hours, and for the 7 hours, no, rest of the 7 hours you simply do other works, like you socialize, you eat, you do some exercise. Okay, if you think that you're going to study for the end, most of the time out of 24 hours, maybe 18 hours you're studying, okay, and you're going to qualify CSIR net, my dear students, again, this is not going to work. Because remember, you are studying, you are actually, it is the brain which is working, right? You have to give some rest to the brain also, so that it gets the time to digest what you have given. Okay, it has to separate the topics, it has to analyze the topic. Okay, so when you're studying, try to study for the maximum time, that is 9 o'clock. 8 hours sleep is definitely required, a minimum of 6 hours is mandatory. Okay, because sleep, you might think that it is a waste of time, but again, it is not a waste of time, it is scientifically proven. This is the time when your brain is doing many work. Okay, and then 7 hours of some sleeping, eating and all the socializing. Now, quickly, I will just go through the important parts. Okay, these are the sub parts under each of the topics, because the details of this will be shared to you if you stay till the end. The detailed topic list. Okay, so for unit 7, go through blood and circulation, cardiovascular system, respiratory system, nervous system, sense organs, excretory system, and endocrine system. Okay. Uh, for some topics you have to prepare the entire topic, for some topic only few subtopics are sufficient. Again, the list of it you will get it at the end of the webinar. For unit 8, Mendelian genetics, gene mapping methods, microbial genetics, human genetics, mutations, structural and numerical alterations, these are the important topics. From unit 9, principle and method of taxonomy, outline classification of plants and animals, and microorganism of conservation concern, these are important. From unit 10, habitat and niche, population ecology, species interaction, community ecology, ecological succession, ecosystem ecology, biogeography, and ecological rules. Okay. Now, all of these are actually the subparts of unit 10, and ecological rules, this comes under subpart A. Okay. So even if you leave out subpart A, just go through this one particular topic. Unit 11, Emergence of evolutionary thoughts, paleontological and evolutionary history, molecular evolution, the mechanism, brain behavior, and evolution. Okay. Unit 12, tissue and cell culture methods, transgenic animals, and gene therapy. These are the important subparts under these units. And lastly, Unit 13, definitely go through molecular biology and recombination method. Subpart histochemical and immunotechniques, biophysical methods, statistical methods, microscopic techniques, electrophysiological methods, and methods in field biology. Okay. Now I'll just quickly show you some 2-3 samples from these few of these units. Like for unit 13, I told you comparative study is very effective, right? So you can definitely use such kind of table. So this is a table which is actually showing you the radioisotope decay process. Okay, and this comes under radio labeling techniques. So what are the different decay and what is the property? You can definitely go through such tables to memorize the differences. For unit 9 classification is a big unit. Okay, and many people, they are very scared of the classification part. But what I will tell you is, just open the book. In the book you will get to see these kinds of mind maps or the flow chart kind of pictures. Go through that. Okay, here only many important points regarding each of the families, each of the classes, they have been given for both plants and animals. This is an example of the classification by Smith and Smith. This is for your pteridophyte. Okay, under plant kingdom. But like this you will get it for your animal kingdom also. Okay, and the rest of the plant kingdom also. So even if you study these charts, they are very effective in memorizing the differences. And for unit 12, like for your the gene transgenic animals and transgenic plants, you can prepare some tables like this. Okay, where you enlist all the list of the genes and what are their functions. Okay, and like for example, in the case of your agrobacterium mediated gene transformation, you can get hold of such kind of picture. Again, these pictures are available in your textbooks. So you can get study this picture. This picture is diagrammatic as well as is in a flow chart kind of, where they have given you step by step what is happening, with genes are functioning and everything. Okay, so such kind of pictures itself, they are self explanatory. Okay, and they give you a lot of details about the chapter. Fine. And as I told you, for unit 11, mnemonic, okay, that is a part where students find it very difficult to memorize the paleontological errors and epochs and all. So use the mnemonic. Like for example, here you have Cambrian, Orthovisian, Silurian, Devonian, Carboniferous, Permian, Triassic, Jurassic, and Cretaceous, right? So how do you memorize that? You use this word creak. Okay, what it stands? Camels often sit down carefully, perhaps their joints creak. Okay, so camel for Cambrium, uh, O for Orthovisian, S for Silurian, okay, D for Devonians, CA for Carboniferous, P for Permian, T for Triassic, J for Jurassic, and C for Cretaceous. Okay, try to get hold of these mnemonics. You can make it yourself as well. Okay, and in that way you will be able to prepare that paleontological uh, time periods. Okay, so these are the different techniques you can use it. So start studying, and definitely with the help of these kinds of techniques, okay, I'm sure you will be able to cover all the 100 important topics for in this 100 days of your preparation. So with that, I will end my session. Thank you very much. I will now pass on the control to our next speaker, Urmi ma'am, right? So wish you all the best. Thank you.
Very good evening, everyone. Am I audible to you all? Please confirm if my slide is visible to you. It's visible to you, right? Okay. Now here we are with session 3, which are the test taking strategies. All throughout, you have heard everything, what you need to do. I will emphasize in my session what is the importance of tests and what you're supposed to do in your main exam. Today's contents will be first, we'll be exploring effective strategies for tackling MCQs, because all you have is MCQs. All you have is MCQs, and which are the most effective strategies, how you can get maximum scoring from there. And most importantly, how you can answer correctly, accurately. Second will be reverse preparation strategies, which are the preparations according to CSIR question types. This is a very new thing. First, you look at the question types and then you prepare through your units accordingly. Third will be smart question selection strategies in part B and C. This is applicable during your exam, but there is no harm in knowing so that your preparation deals around with it. What type of questions you should attempt, what types you should avoid. The last one will be how important is the practice and mock tests, and from where you should do, how often you should. Let's begin with the first part, exploring effective strategies for tackling MCQs. First is read the questions carefully, eliminate the incorrect options, answer the question without looking at the options, and read every answer option. Let me elaborate each one of them. The first one, read the questions carefully. Make sure you understand what the question is asking. And as you read, you have to focus on important words that change the meaning of a sentence completely, like not, always, except. They could easily trip you up. Many students choose the answer that is the opposite of the correct response simply because they miss a word or they have misread the question. Example, when they are asking in select all the correct answers or select all the incorrect answers, or simply they forget what the question is actually asking. The second is elimination method. Read each of the question, read each of the answers and eliminate the ones that are wrong. They may not be related to the question, or they may be completely off. And then you will have some remaining options. Read the question again and try eliminating which options are not applicable. It's okay to restart the question if you think you do not have the correct answer. Ideally, you can do it in 2 rounds. During the first round, eliminate anything that is flat out wrong, the answers that contain false information or which are irrelevant to the question. And the second one is, you will have to see, before you obsess over the remaining answer choices, read it the question again and remind yourself what you're looking in a correct response. This is when you start your second round of elimination. And then you can compare the remaining responses. Third is, answer the question without looking at the options. This is a very effective method. Trust me, students. It will reduce visual distractions for you. When you look at the question as a whole in the computer screen, it will help you avoid falling into a trap, because the moment your brain sees any wrong statement, it kind of, your brain is a camera, it takes a picture, and that might come into the way of you getting to the right answer. So, coming up with the answer yourself without looking at the options will help you identify what the correct option should say, and that too quickly. That's why compare your answer to the options, look for the same ideas and key terms, think about the right answer, how it would say or contain,
and use that ideal answer as a measure, yardstick to measure the available choices. And the last one is read every option, read every answer option. So the thing is, before choosing a final option, there might be a mistake which students most often do, that they will quickly assume that they know the correct answer. But don't do that, because there might be a best answer to some of the questions. And that is why, without first reading every answer option, do not try to give the right answer. Okay. I'll give you a bonus tip here. Look for common types of wrong answer options. That means look for a pattern. Learn to recognize frequently used option patterns. See, your question paper setters are also will frame a question in which there will be some wrong choices, isn't it? So that would be hard on them as well. That's why they will try to formulate wrong but attractive choices. So if you learn to recognize these frequently used options, it will be easier for you to eliminate. I'll give you some examples. Some of the patterns, the wrong options which you will have, these are, they will include reversal. Okay, reversal means the answer may feature an error in chronology, in the order of a sequence. There might be a difference, or characterizing the first event as the second, or a detail that is the exact opposite of the truth. Some things which is true but irrelevant. The information might be correct, but it is not applicable to the question. This you will find generally in experimental type of questions. Too extreme, the answer takes a fact that is true, but it might exaggerate it to an extreme with usage of some words. For example, it will insist that it is always true, when in reality it may be only sometimes true. And the other one is just getting one detail wrong. This is a favorite choice of CSIR question paper setters. They will add a word not, or they will add a word without, with to without, presence to absence. So with just getting one detail wrong, the answer, entire statement, sentence might seem perfect, except for that one little detail which you might lose out on seeing it. So you have to focus on every word. Coming to the second part of my lecture, reverse preparation strategy. So this is a very interesting concept. Preparation according to CSIR question types. How is it going to help you? So first of all, telling you the question types that you have, you will get concept based, match the following, graph, diagram or pathway based questions, numerical type or formula based questions, arranging or ordering the questions, and aptitude. So first, talking about concept based questions. How will you prepare for these type of questions? Now when you are preparing for your during your preparation time, let's say tomorrow you sit with a unit and you come across a certain concept such as replication, let's say. So you will remember all these 6 categories of the questions, and then accordingly you will prepare. So whenever you look at a topic, for example this replication, as I said, immediately you will think that some concept can come from here. How will you prepare? It should ring in your bell. So remember these points very carefully. You have to do in-depth study of the topic, preferably from any standard reference book, because concept is always something which stays with you for a longer time. If you memorize something, that might you miss out one or two things from there, but a concept will still be there. If you see a movie, you will remember the story, what the concept of the movie was. Probably you will forget some scenes. Then write down short summary points of each concept. As Somrhita ma'am elaborated, you write down the bullet points or however ma'am has explained to you. To study the concepts, you have to write very concise ones. Don't write long sentences. And use recall method to revise. How to revise concepts if it is an entire concept? Definitely you can look at the summary points. Since you have 100 days only, you have to be very quick with your preparation. So once you make this short summary notes, when you're revising, just close the book. Okay, I am supposed to revise replication. In that proprietary replication, you've written down some points. Close the book and try to recall what all points are there. And then you can open the book and see which point you have missed out. Very, very important. Do follow this. Coming to the next one, match the following questions. Always make color coded tables for such kind of questions and study. What are the examples? Enzymes, cofactors. Genes, their functions. Mutation, what will be the phenotypic effect? Or anything under the sun. What technique, which of the unit 13 technique, what exactly it does. Okay, and just now as Shramita ma'am had shown, what are the weird genes in agrobacteria mediated gene transfer, what is their function. So always make color coded. Color coded gives you a visual impact. You can also refer to Biotechnica's ready-made comparative tables, which are concept tables and concept wheels. Those are very good revision aids when you have less time in your hand to study. Choose the rows and columns wisely. What points you are supposed to enter in your table. For example, if you have the microfilaments, actin, myosin, or even some motor proteins, kinesin, so your rows will be obviously the type of the microfilament or the cytoskeletal element. And then second will be column will be the function. In that same row, in that same table, you can include a third row, which will be the inhibitors or the ones which are inhibiting in polymerization or ones which are inhibiting in depolymerization. So like that you can keep on including. For this, don't think that at once you can make these tables. No, you have to maintain a book, so that in the book you can make the tables and rows, or you can just keep loose sheets and in a file, and in that file you keep on entering these sheets as and when they get filled up, and you have to keep extra space for adding up the points as and when you are studying. Very important. Coming to the next one, graph, diagram, and pathway based questions. These always should be studied through something visual interpretations. What will you do for that? You can take some colored printouts or you can draw them, but since you have less time less left, so I will prefer to take some printouts, colored printouts, or else take black and white printout and in that highlight important terms and points. And most important thing is that they should be easily accessible. So either you can carry it around with you in a file or you can stick it in your room where you can see it all the time. This is how these will get stuck in your head. For example, the graph of, let's say, protein folding, or the graph of or the diagram of cell signaling, or some metabolic pathway. So these you just cannot uh, open the books every time and revise. So better to have these kind of images to be carried along with you. Next is numerical type formula based questions. Write the topic name and all the formulas related to it. Pick up anything, let's say Hardy-Weinberg equilibrium. Write down all the formulas that will be applicable and in all different formats. Similarly, enzyme kinetics or anything from cell biology, right? Elaborate abbreviations. So let's say you are writing equation. What is KCAT stands for? What does KCAT stand for? What does KM stand for? So you write the formula and at the bottom you write where this is that. And add an additional note on the required unit conversion methods and other miscellaneous factors. For example, in thermodynamics, if you're solving questions or pH buffer, you will require log rules. Also, the conversion methods like micrometer to nanometer, whether you have to multiply by 100 or 1000 or divide by 1000, everything you should make a note of it. Don't forget, everything should be very concise. Coming to the next one, arranging or ordering questions. Usually it is applicable for a process or technique. For example, if you are doing transgenic plants or animals, or you are studying any technique in Unit 13, FSES, then what are the steps that you do or follow in that? Here, flowcharts are very helpful. So you can draw clear and concise flowcharts step by step. They will ask you to arrange them in order of their sequence and add an image or diagram for better understanding. Sometimes you don't even have to draw the flowchart as the agrobacteria mediated diagram Somrhita ma'am showed. And sometimes the diagram can be itself explanatory. For example, the in unit 6 plant biology, PS1, PS2. What are the carriers, electron carriers in between? So these all you can keep it and study accordingly. I hope you are noting down while you study these topics. You should immediately think of the type of question that can come in the paper and accordingly prepare. This will help you prepare faster in less time. And the last is aptitude section. which you can't really memorize or study. This is something which you develop. So, first, jot down all the formulas, solving methods for each topic, and also practice at least 10 questions every day. That is how you will develop. Later, you can do something when you have mastered the accuracy. Once you're getting everything correctly, then you can work on the speed. Now, the third element of my session, smart question selection strategies in part B and C. Let's dig in. So, the first one is selection of the questions. What type of questions you should answer? Direct knowledge based questions. For example, these. Which of the following is a foodborne toxin? How much energy is produced in TCA cycle? Which out of this has this? Use the two component histidine kinase receptor. So these either you know or you don't know. Provided you have prepared these units, definitely go for these direct questions because they are definitely these are from part B. So they are very easy and quickly you can solve them. It will save time for you. Then try to go for some match the following questions and more graph or diagram based questions because there is less chance that you can get them wrong. For example, this kind of match the following questions. This is a no brainer. I'll come to more match the following details. Then easy option questions. Now, this is not an easy option question. This is an example of an easy option question. But if you think that this is a lengthy question from that point of view, yes, I will come to another example where I will say avoid lengthy questions, but just give a close look, just a scanning of the question, that what it is happening. If you see some opposing statements, for example, if you see here D glycerol, L glycerol, rest everything is same. Then again here L glycerol, D glycerol, rest everything is same. Ester bonds, ether bonds. Rest everything is same. So like that, opposing statements are very easy to solve. So that is why these are easy combinations. It might look very big. Chances are that many students might skip it. But if you correctly know the answer, you can solve it under 1 minute. Shorter introduction part C questions. Go for the ones which quickly can be processed in your head. Okay. And lastly, similar sentence options. Just now, which I showed you, similar sentence. Okay. So this is one more thing. Avoid multiple table match the following questions. While I told you to go for match the following questions, but just have a look at this question. There are 4 tables you should match, not just one. Okay. And just have a look at the options. How confusing is that? When you are already stressed in a 3-hour exam, for me, this is absolute no, even if you have studied from it. Or else you can keep it at the end. If you have studied sex determination properly, keep it at the end. Do not jumble up your head right away. Okay. Now I will show you some more part B questions. Have a look at this question. This is quite big for a part B question. See, part B questions are supposed to be quick, solved quickly. Okay. Major chunk of the exam you are supposed to save for part C. So, my suggestion is avoid long sentence part B questions. It will be more time consuming, and to read these sentences and process an answer, it will involve thinking and processing. Okay? Please do not go by the subject of the question because I am telling in general what type of questions you should select. Obviously, if you have prepared developmental biology, you will go for that question. That is a different thing. But if you have another alternative, that you have a direct part B question from developmental, solve that first. You can come back to these later. Okay. Now you see this part B question. By the way, it is from the same exam in which you had that question. This question was also there. I will suggest go for these type of questions. Attempt short direct part B questions, saves time for part C and no brainer to solve. Most important, it gives you confidence. Another example for such kind of part B, easy part B, just one word, one sentence, easy to process. Go for such questions. Coming to part C question, have a look at the length of this part C question, and length is not important. There is a table involved in this question, there is a diagram involved in this question, there is a explanation involved in this question. To at least understand this question, it is going to take you minimum 2-3 minutes. Solving time is separate. So even if you have studied from here, it will be a bad luck if such lengthy questions come from your area of studies. So my 2 cents for this is avoid complex part C questions like this. You can see another one. This is from unit 13 to find the sequence of the protein. So many treatments are there. Okay, so many treatments, individual fragments. You have to join the fragments, get the sequence, and then find the disulfide linkage. Minimum 10 minutes it will take for this question. Strictly no. At least keep it for the end. Remember, somehow you have to get your 25 questions done, part C. So focus on that. This is one more part C question. Now see, here there are no options actually. These are only the options. So it's a direct question. In part C, this will be considered a shorter length question. So which are having simpler sentence based questions. Choosing of the sentences. This will be comparatively easy, as I showed you the previous picture with the diagram and table. So being a part C, this will take time, this question, but at least not more than 5 minutes. Now, questions having opposing statements can also be easy sometimes. Now, this is also a part B question. As I said, these two is opposing statement. Either one of this is correct. These two are opposing statement. Either one of this is correct. Being a part C question, hardly it will take you 2-3 minutes. Not even 2-3 minutes. 2 minutes is maximum, provided you know this concept. And this is a very common concept. If you have a decent preparation of biochemistry, thermodynamics, this is a decent topic, you will get it right. I will show you one more. Choose the answer, correct options. Look at the options which are given. First one is all the 4 options. Second one is 3 options. Third one is 2 options, and last one is 1 option. These are very risky type of questions. Okay, so you can have 2 also correct. The previous one which I showed you, even so you can see this as avoid questions with complex pattern of options. But as you see this one, so either 2 will be wrong, and for these type of question, elimination works very well. Okay, so the ones which are straight away wrong, you can directly cancel. You can at least narrow down your options. So these are more easy choice of questions. The last part, coming to the practice and mock tests, tips for a rock solid preparation. You should always revise on a daily and weekly basis. Ideally, what you are studying, you should revise it thrice before the exam. If not thrice, at least try twice. So it's about revising little but often. My colleague, Dr. Tanushri, will be throwing more light on this part. Now, next is practice tests. Practice tests, previous year question paper, mock tests, other exam paper questions. DBT, GATE, ICMR, select anything. And as Somrhita ma'am said, questions at the back of the reference book chapter. So these are the ones which are quite analytical, and it will help you in CSI preparation. So what is a good source for practicing questions? Previous year question paper? Yes. Questions at the back of chapter? Also yes. But what you're not getting over there is the proper evaluation or the detailed explanations, regularity, who is monitoring you whether you're doing or not, and you're not getting unit wise questions. So what is the solution? You can opt for something, a platform where they are providing weekly practice tests, like how we do at Biotechnica, or monthly mock quests. So whatever we have learned in a month, it will be in the pattern of the CSIR exam, and you can find out how and where you are standing. AIMNet test series, which is AI powered, it is giving you all the unit wise. If you have studied entire of unit 2 and we want to see whether you are able to solve questions, you can go for AIMNet. ToughNet for more tougher questions from part C if you want to challenge yourself. The best part is that what you should do after taking test. This is what most people miss out. Nobody is going to tell you about that. So first thing is postmortem of the question and answer. Yes. Find out, get the detailed explanation of the question, and ensure you know all concepts which are related to the question and answer. For example, if you are studying Drosophila and they have given you something regarding the axis formation, then why not study the axis formation in amphibians also and sea urchin also at the same time? Work on 3 R's. First is rectify. What mistakes you have done? You have just solved a numerical, but you forgot the unit conversion. Maintain record of that. That this was a mistake, common mistake I'm doing in my tests. Trust me, if you do all this, nobody can stop you from clearing CSR in 100 days. Refine. Ensure to take steps not to repeat same mistake. So whatever record you have kept, keep seeing it time to time. So it will make an alert in your brain not to repeat those mistakes. Reforge. Remodel concepts in your brain. When you have made a mistake, that means somewhere in the brain that concept is there. Throw it out and enter a new concept in your brain, the corrected one. Okay. So as I said, try to have an online platform for practice tests. For example, in Biotechnica's weekly tests and quests, this is the format which we have, online format. And after the question, the vivid explanations are also given, which helps you to understand the concept behind the question. And best part is that it has a interface, the practice for practicing question, it is same as that of the NTA platform. So you're already practiced to appear in the exam. If you are not well versed with giving an online test, then you will be in the proper course. I believe this was helpful to you. Be a game changer. The world is already full of players. Do something challenging, and then only you will be successful. Thank you for your attention. I pass on the controls to my next speaker. Thank you, everyone.
Yeah. Am I audible to everyone? Still no voice? Can you all hear me? It's fine. Okay. Thank you so much, Rimi, for confirming. I really apologize for the inconvenience. So I was saying, I was just telling you that this session is all about time management. And I was saying that I'm sure you all will agree that the time management is not only important during the CSIR NET exam, but it is also very much important during the entire CSIR NET exam preparation period. So if you are worried about that, I have only 100 days, how will I complete my syllabus? How will I complete the topics? How will I revise? So don't worry. In this session, I will let you know about certain tips time management tips that will help you prepare effectively for CSIR NET exam in this 100 days period. Remember, everything is achievable by proper planning and time management. So this should be your approach for 100 days preparation of CSIR NET exam. Now, before talking about the tips, before letting you know about the tips, time management tips, I would like to tell you all about that. What is the importance of time management in CSIR NET Life Science 100 days preparation? So very first importance is that the effective time management helps you cover syllabus, helps you cover extensive syllabus. There are total 13 units in the CSIR NET exam syllabus, although you need not to prepare all, but 7 to 8 units also if you are preparing, then every unit has several subunits and topics in it, right? So effective time management ensures that you allocate sufficient time to cover all the topics comprehensively within the given time frame. The second importance is that the time management ensures balancing multiple subjects and unit. Tell me, does it happen with you that you devote too much time on one subject? Like when you study if you go through the syllabus of CSIR NET exam, there are many subjects, right? Like biochemistry, genetics. Unit 8 is all about genetics. Unit 1 is about biochemistry. Unit 13 is about techniques. Unit 3, Unit 4, cell biology, molecular biology, right? Now, does it happen with you that you plan, but you devote too much time on one subject, and at the end, you have no time left for studying another subject or no time for revision? Tell me in the chat, does it happen with you? Yes? I'm sure those who have already prepared or are in a phase of preparing, it must be happening with you all, right? So the importance of effective time management is that it allows you to allocate time to each subject based on the weightage in the exam and proficiency level. Then it also helps you strategize preparation approach. Like you can prioritize the topics based on their importance, and even you can devote more time to the topics you find challenging or the units which you find challenging, and maybe you can devote less time to the easier ones. So it helps you strategize preparation approach as well. Then it helps you schedule the regular practice and revision sessions, which you all know is very, very crucial for CSIR NET exam. Then even it helps you optimize productivity and efficiency. So basically it helps you maintain balance between study, rest, and other activities. And it also ensures that you make most of your study hours by staying focused and minimizing distractions. So this way it helps you optimize productivity and efficiency. Then even it helps reduce stress, last minute stress and anxiety, which happens just because you are not able to complete the syllabus or due to lack of revision. So stress management, effective time management helps you in stress management. And by following the time management, effective time management, you can achieve your target, you can complete the syllabus, your revision part, your practicing question part, and therefore it will give you a sense of achievement and even boost your confidence, which is very, very much needed for CSIR NET exam. Now the question arises how to manage time. I told you about it is important, but the biggest question is how to manage it, how to manage the time during this 100 days, the time for 100 days period for CSIR NET preparation. So very first thing is you have to create a personalized 100 days study schedule. See, everyone has their own, you all must be having different schedules. Like somebody is in the job and also preparing for CSIR NET, somebody is attending the colleges, and some are simply preparing for CSIR NET exam. So you have to prepare a personalized study schedule and make sure that when you are preparing a study schedule, you plan. You plan the month, week, and a day. So you have to allocate the time for the units, the subunits, and the topics you are going to do in a day or in a week or a month. And make sure that when you plan, you set realistic goals. See, everyone knows that how much you can study. Some are fast learners, some are slow learners, right? Some are medium, like average learners. So you have to understand that if you are keeping a target, like I will be completing 8 to 10 topics in a day or 2 subunits in a day, right? It's not possible because every subunit has also many topics in it, right? So set realistic goals. I'll suggest that don't go for more than 2 subunits of or 2 units in a day. Okay. Then even when you plan, you have to allocate specific time slots for different units and topics. Don't study one unit for entire day. Don't do like this that I'm studying biochemistry for the entire day. That is the wrong approach. You have to bring variation in your study. And for that, what you can do is like you can select one easy unit, which the unit which you find easy, and one difficult unit. And you can select topics from each and you can prepare them. So you have to allocate specific time slots for different units and topics, and revision is very much needed. So you have to allocate time for revision as well as question practice, because at the end, at the time of exam, you have to practice, you have to solve the questions only. Then maintain balance between study, rest, and other activities. During these 100 days, physical and mental health, your physical and mental health is very, very much important. So make sure that you maintain a balance between the study, the rest, and other activities, curricular activities that you are doing. And tracking is very much needed. Tracking of the progress. Sometimes it happens we plan and we are not able to cover that, some maybe due to some unforeseen circumstances we are not able to complete a particular topic. Let's say you plan to study photosynthesis and you're not able to cover that. So don't just keep on thinking about it or just feel demotivated. Just track your progress, what you have planned, and whether you are able to achieve it or not. And if something is missed, then adjust your timetable as needed. So I'll just show you a sample of your 100 day study plan. So for a week I'm showing it to you. So like you can mention the date, you can mention the day. And the very important thing here to note is that keep some your morning time, like when you wake up, some time for physical activity, 15 to 20 minutes at least. Like either you can go for a walk, or you can go for do some exercise or yoga or meditation. This is very much needed to keep you active throughout the day. Now let's say I selected unit 1 and unit 6. So I told you have to select 2 units. So assign day slots for units in a week, and also you have to assign hour slots for subunits in a day. Now, let's say, like here I have mentioned the subunit, but you can mention the topic, like let's say you are studying hydrogen bonding. So always start with important topics from that particular unit or subunit. So I want to study hydrogen bonding. Now, you can allocate 2 to 3 hours for this. Now, make sure 2 to 3 hours doesn't mean you have to continuously study about hydrogen bonding for continuously for 2 or 3 hours. No. What you can do is like for 45 minutes, continuous you study about hydrogen bonding, all the important points. Then you can take 5 to 10 minutes break. So in first 45 minutes you study and you understand the topic, right, the concept. Then you take a break, and after that what you can do is you can like either you can take up like you can make notes on that particular topic. So make notes on hydrogen bonding, right? So this is how you can revise. Also you can use a method where without seeing the book, like you just write down on the paper what you have studied about the hydrogen bonding, and then once you're done with it, open the book and see which point you have missed, and you can add them in your notebook. So this way you can make notes. Then you have to break, like you have to devote, like when you are making a plan, you have to include the time for break. Okay, you don't have to study continuously, otherwise you will get tired and bored. So like in morning, let's say you study about hydrogen bonding, so in the afternoon you can, let's say, study about hydrophobic interactions or van der Waals forces. Now between these two study sessions, you can practice questions maybe based on the topic that you have studied previous day, or maybe on the same topic, like you can practice questions on hydrogen bonding itself. Then again, after the second afternoon session, you have to take a break. Now during the, like in this plan, you have to make sure that you have to devote time for general aptitude also. So like 5 to 10 questions you can solve from general aptitude. So study a concept. Let's say you are solving probability questions or questions based on determining the ages, or any number series questions you are solving. So first understand the concept, go through the concept, and then practice the questions based on it. So 5 to 10 questions or 8 to 10 questions you can practice every day. And then in the evening, like, you can study photosynthesis. So you can study about dark reaction, light reaction. And finally, at the night, you can practice or do the revision, right? You can revise what you have studied in the studied morning, or even you can practice the questions from the topic you have completed in morning. And before going to bed, plan for the next day, and always, like, mark whether you are able to achieve the target or not. If something is missed, don't worry, keep Sunday only, or Saturday Sunday as a buffer day where you can complete any pending topic. And most importantly, you have to give the mock tests and practice more questions from previous year papers. Make sure that you devote every day self study 8 to 10, like every day self study of 8 to 10 hours is must during these 100 days. So the complete 100 days study plan, like, we have prepared a complete 100 days study plan for you all. So the link will be shared in the chat, and you can download the PDF of that study plan from there. Now, the biggest problem of planning is that we often forget. We plan, but we forget that we have to follow a plan, or we have to do a particular task after once this task is over. So for that, you can take help of several apps, and the apps here I have shown on you can see it on your screen. So, like, you can use any.do, you can use Google Task Manager. So all you need to do is you can set your task, and like, you can mention the task and set a reminder. So these apps will give you a reminder about the task. Like in Google Calendar, even you can mention your study, you can study sessions you can mark. And Amazon Alexa, you can use it for like to set an alarm or for reminder. And even you can schedule an email to you. Like, you can set a task and email it to yourself, schedule an email to yourself. Like, maybe like today you wrote down some task and schedule it for next day, like what topic or unit you want to study, so you can schedule it for another day. Now, even while note making, you can take help of apps like Otter.ai or Zoho Notebook. So basically, like, let's say when you are attending the lectures, so Otter.ai transcribe the voice into text. So this way you can make notes without noting anything. And when you have the notes made through this Otter.ai and the audio recordings, you can use it later for revision. You can go through them for the revision purposes later. Even you can use Zoho Notebook for making the notes. Then even Pomodoro timer app is there, which you can use for managing the time, and it will allow you to focus on your task, that is study. Then one more thing I would like to tell you all here is that please note these 100 days are very much crucial for you, right? So at this time, you have to stay away from distractions. So if you are in a habit of, if you are devoting too much time on your mobile phones or on any social media platform, then you can take help of these apps, Stay Free and Stay Focused. Stay Free app, it basically record your app usage so that you can know that in which app you have devoted too much time, and so you can control your time wasting habits using this app. And Stay Focused, it will allow you to block any app or site. Then even you can use Headspace. So at this, as I told you, that during this 100 days, physical and mental health is very much important. Maybe during this phase you feel demotivated, you feel stressed. In that case, you can take help of Headspace app. So Headspace app is basically a meditation and mindfulness app. So you can use it to improve your memory, improve your focus, and to remain stress free. And lastly, but most importantly, you can always connect. You can download the Biotechnica app and connect to us on our WhatsApp and Telegram group. So it's a one-stop destination for all your biotech and life science needs. You will get the notifications related to the exams, about the scholarships, about the internships. And even if you sometimes feel demotivated or you need guidance in solving some question or in studying to understand any topic, you can always reach out to us through our WhatsApp channel and this Telegram group. Then lastly, I would like to tell you about the time management in question solving. Tell me that does it happen with you that when you are giving the mock test, or maybe some of you have given the CSIR NET exam earlier, does it happen with you that you devote too much time on one section, so at the end the time left for another section, like you get no time for solving the questions of another section? Please let me know in the chat. Does it happen with you? Yes, I'm sure some of you will agree with me that it is quite possible, right? So this happens when we don't follow time management in question solving. So here are some tips that you can follow for time management in question solving. First is set deadline for each section. Part C like you need to devote more time in part C, because part C questions are difficult, lengthy, as shown by Urmi ma'am and in the previous section and previous session, and also like they are experimental based questions and need analytical skills, right? So you can devote 1.5 hour for part C, 1 hour for part B, and 20 minutes for part A, and last 10 minutes you can keep it to go through entire paper. Now, even when you are practicing the questions, you have to allocate the time for each question as well. So since we are devoting 1.5 hour for part C, so 3.6 minute, like you don't have to spend more than 3.6 minutes for each question, part C question. So 25 questions you have to do, and in part B 35 questions. So you should not spend more than 1.7 minutes for each section. Stick to time allocation to ensure that you complete all the questions within the stipulated time. Then also I will suggest that you try with solving those questions first which you find easy, like part C questions. Even if you're solving, start with those which you find easy, like match the following type questions are there, right? So you can solve those questions. The reason is that when you start with easy questions, you'll get more of time to attempt moderate or difficult questions. And when you start with easy questions, it increases your confidence, which is very much needed during the exam. And you can leave the questions that you're uncertain about for the end, like you can mark it for review. So the point is, don't get stuck when you are practicing the questions, and don't hesitate to seek support. Remember, we are always there for you. So don't hesitate in seeking support, be it you need it for practicing questions, and you need help in solving questions, or understanding a particular topic. So always reach out to us. So the takeaway message is, remember, time management is a skill and it developed with practice. So this 100 days CSIR NET preparation mantra is: be consistent with question practice, give time-bound mock test, and adhere to structured study schedule. This way, you will enhance your time management skills, and definitely it will improve your performance in CSIR NET exam. So I'm sure you find this session helpful. I've already told you about time management tips. Please remember, knowing is not enough. You must execute. So this is all about time management. Now it's time for you all to know about the revision strategies. So which will be taken by Dr. Violet ma'am. So over to you, Dr. Violet. Thank you, everyone.
Good evening, everyone. Please confirm if I am audible and if the slides are visible. Okay. So today I am here to discuss about the revision strategy to adopt in the next 100 days. So till now we have been listening to so many tips and tricks that you need to follow in the 100 days. But what is most important is to be a smart learner, right? We cannot just be a rote learner. We just cannot keep on studying the notes, studying the textbooks, right? The PPTs. We have to, we have just 100 days. So we have to be a smart learner so that in those 100 days, you are prepared well enough so that you clear the CSIR NET exam, right? So what you need to do to be a smart learner? So I'll be sharing some of the So make sure that you be a smart learner. That will help you to be a holistic learner, that is a complete learner, just not studying from the textbooks, and it will help you in increasing your engagement. So whatever you study, whatever you are studying, so use these techniques and tricks that we are going to share over here, though some of it you have already seen in the previous sessions when we were discussing about the various topics that, in the unit wise, what all needs to be followed. So whenever you are studying, first use the flow charts. Okay, so in flow charts, like I have shown you one example over here from unit 7. So it is a topic that is about our AA system, right? So you can see there are so many different steps. There are around 16 steps. But if you follow studying through the flow charts instead of going through the textbook, instead of just reading through the textbook and through the PPTs, so it will be wastage of time. But when you follow these flow charts, it will be very easy, and in a very less time you can you can understand the concept as well as you can remember it. So remember, whenever you are studying, follow the flow charts. You will get these flow charts, in the various textbooks. Even you can prepare these flow charts, as well as I will share, like Biotechnica has certain Conceptica. So those you can use for studying these flow charts. That will help you to study a lot of topics in very less time. Next is, always remember to go through the figures, be it immunology, be it, I've given an example that you can see for biochemistry, or you can, talk about unit 2, that is cellular biology. So any topic that you talk about, okay, you can use these figures that will help you to understand and remember the concepts in very less time, right? So use these figures. Like here you can see this is an example of how the proteins are being transported inside the mitochondria, as well as this figure is for the proteins, how they are being transported inside the chloroplast. So you can see, just by looking at the figure, you can know what are the different transporters that are available, how the protein is moving inside, whether it is being cleaved or not getting cleaved. Okay, so all these steps can be easily understood and you can easily remember. So this is very important, that whenever you are studying, go through the figures, do not miss the figures. And this way you are, developing the habit of being a smart learner. Next is, always use mnemonics. So, what is mnemonics? Basically, it is a technique that you can use to remember the important points related to some, you can see over here example where you need to know about the different hormones that are released by anterior pituitary. Now, here what happens, you might forget, right? There are so many different hormones. So, if you use the mnemonics where you can, it is basically the first letter of that particular word. So, you can use it to form a particular pattern and you can remember it like this, like flat pig. So, it is for FSH, LH, ACTH, TSH, prolactin, and GH. So, in this way, you can use mnemonics to remember difficult topics, okay, difficult terms, difficult examples, okay, where you have a lot of things to remember in a very less time. Another example, you can see essential amino acids. So math will apply. So M is for methionine, A for arginine. So like this, you can easily remember so many different, around 10 amino acids over here with just one word here. So you can use them in mnemonics. You will get it, for different topics. Usually, you can get it online, or what you can do is you can even prepare your own mnemonics. So it is always better when you prepare your own mnemonics, because that will help you to remember better. So this is the chart that I was talking about. So Biotechnica especially, has certain smart learning tools. So one of them is Conceptica Lite, where you can see a topic that is cancer. That is one of the important topics, and you need to know so much about cancer. You need to know what are the difference between normal cells, what is a cancer cell, what are the, types of tumor, what are their characteristics, and then what are the different types of cancer like carcinoma, sarcoma, what are those. Even, you might have found that questions are asked from here, right? Either it can be part B or part C. So you can see, just in one chart, all the important details have been written, and it is very easy, it is very attractive, where you can easily remember. It's colorful, right? So it will easily help you to memorize things better. It helps in memory retention, quick recalling, right? So that is why it is always important that whenever you study, you use these concept charts. Another example that you can see over here is from unit 7. So it is based on the same concept charts known as conceptica. So here, like some of the important topics, like you see, they ask questions about the year. So the different parts of the year and all about, the quick notes which you can easily study and remember, so that in exam you can easily recall. So use these concept charts when you are preparing, when you are revising, right? Next, another example you can see from unit 2, where you need to know about microtubule depolymerization, and another topic is dynamic instability of the microtubules. So for that, you need to go through the textbook, right? We usually refer CARP, right? But if you follow this chart, so you have the figure, you know what is happening, and then you have the different steps, okay, how it is occurring. So it is in very less time you can easily remember the difficult concepts, the difficult topics. So always prefer these concept charts whenever you are studying for and preparing for CSIR NET exam, and especially when you have just 100 days. Now another important way to be a smart learner is to study by using mind maps. Mind maps helps for easy interpretation. So what is mind map? So mind map is something like you have a topic, and then there are so many things that come under it, right? And you connect all of them together, right? If you see here, if you need to know about the function of nephrons and collecting duct, now nephrons have different parts, right? It has PCT, it has DCT, early DCT, late DCT, nephron loop. Okay, then the beginning part, that is the renal corpuscle. So there are so many different parts that are just present in nephrons, and each part of the nephron has a different function. So what you need to do over here is you need to make mind maps. So this is there are some tools that you can use for preparing the mind maps, as you can see one of the examples, right? That's a e-draw tool that you can use. Apart from that, you can also see these figure based mind maps. Okay, so these are also very no, very colorful and very interesting, right? So you can easily remember about, so we have so many different endocrine glands, they have different hormones, they have different functions. So if you follow these mind maps, it will help you to remember a difficult topic, a lengthy topic, in a very easy manner. So it will help you to understand things better and to retain better. So that is what is important for preparing for CSIR NET exam. Next, another important strategy when you are doing your revision is use the concept wheels. So, what is concept wheel? So, there are some important topics, right? Today it has been shared with you all. What are the 100 important topics? So, like one of the topics, like if you talk about ion channels or astrocytes, so this wheel is about that particular topic, and the spokes of the wheel are the different characteristics for that particular topic. So in this way, what is happening is, you can see it is the concept wheels, they are very colorful, in different shapes, and it has a central idea that is about the astrocytes or about the ion channels, and each spoke is talking about the characteristic or the important point. Okay, so it is very easy, and it is a very fun and effective way to remember concepts for retention, and it is a kind of visual learning. So these always help, and you never forget these kind of strategies when you use for studying. So it is more of visual learning that you will always remember. Okay, and it is easy for retention. So try following these concept fields. So there are other examples as well from other units, like for cell junctions, then MYC or proto oncogenes. So definitely you will see questions are asked in CSIR from these topics, and you need to know about the MYC family, right? What are the genes involved, what all diseases are caused. So you can see each spoke has some important point, right? So you can easily follow these concept wheels for memorizing the important topics. Another strategy is using the concept tables. Now, the concept tables helps in comparative study. So in comparative study, The concept is in the form of points and tables, as you can see over here. So whenever some topics that you need to compare, like you need to know about vasodilation, vasoconstriction, right? So in that way, you can use, for those topics you can use these concept tables. Now what happens over here, you can see, so they are very engaging and a difficult topic. Like, no, we know what is vasodilation, but we don't know the details of it, right? So it is widening of blood vessels, it is caused by relaxation. So how and why it is caused, and what is the difference of vasodilation with vasoconstriction? Okay, so what is involved over here? It increases the blood flow when it is vasodilation, it will decrease the blood flow when it is vasoconstriction. So which part of the nervous system is involved over here? It is parasympathetic. For vasoconstriction, it is sympathetic. So what is happening is the details of that particular topic, okay, though we do not know the details, but when you follow these tables, you can see these are comparative points that have been written. So not only that particular topic, that is the vasodilation, along with it you're also studying the vasoconstriction part. So this is kind of learning in form of differences, so it will help in longer retention of the knowledge that is needed for studying the various topics, right? And apart from that, there are, these are important topics along with examples. So if you see the difference between fimbriae and flagella, so where you will find it, so those examples are also mentioned. So these are very important, okay, these concept tables when you are doing a comparative study. And with this comparative study, you also know about that particular topic. Another important strategy for revision is using the concept cloud. Now, Biotechnica has come up with this concept cloud idea. So, what is this concept cloud? As you can see over here, so for every unit, we have a concept cloud where there are topics that are mentioned. Like for unit 2, you have so many different topics, right? So, even the important and not so very important topics, all of these topics have been mentioned. And so they vary in the font size, if you see. So the important ones, they are more bigger in font size. Okay, so in this way, what will happen is once when you are studying, okay, and you uh, know when you want to check what all you remember, so you can use these concept clouds. So you can see what all topic you remember, what all you need to go back and check, what all you need to revise. So in this way, the concept clouds will be very helpful for your last minute preparation to check whatever you have studied, is there, so these, all these topics you remember or not. Okay, so likewise for other units also, the concept cloud is there. So these are some of the important the ways of preparing and doing your revision. Now you all might be getting confused, like when to use what. Okay, so lastly, I would like to talk about the strategy. So in these 100 days, how and when to use which type of concept, these revision tools. So first I would say, in the so when we are talking about the 100 days, so like for the first 30 days, you can think like, you will be studying, you will be understanding the concepts, you will be learning about some topics that you have never studied before. So definitely that time use the diagrams and the flow charts, and to remember those topics, use mnemonics. Okay, so in this way you can use, in the very beginning of your preparation, use these diagrams, flow charts and mnemonics. Now once you have studied those topics, next important thing is you need to know about, you need to revise, right? So just studying will not help. What is important is you have to revise. So use the smart revision tools that I spoke about, right? Because already you have studied through the diagrams to the flow charts. Now you need to revise. So use the concept wheels, use conceptica, use concept tables. So this will help you to understand the concept better, okay, and to retain the concept for a longer duration, and to not to be completely, to remember, it will help to remember each and every topic when you are following this strategy. Then finally, in the last 30 days, when you are doing your revision, you are solving previous year question papers, I would suggest go through the concept cloud, because whatever you have studied, okay, in the first 60 days, so that you need to check whether you remember or not. Okay, if anything you have forgotten, so you need to check in the concept cloud whether you have covered all the important topics. If you have missed anything or if you do not remember anything, you need to go back and you need to again study that. So in this way, this strategy, if you follow, I'm sure it will definitely help you in clearing and performing better in CSI net exam. So with this, I would now thank everyone for being such attentive listeners. Now I pass on the controls to Dr. Nupur ma'am. Thank you.
Hello everyone, can you hear me? Am I audible? Let me know if I'm audible. All right. Yes, so I welcome you to Biotechnica's webinar on 100 days preparation strategy for CSIR NET exam. And this is the last session for the day. I'm Dr. Nupur Nagavekar, and I'll be talking about stress management and one of the ways to deal with stress, that is how to practice mindfulness. Right. So, uh, you're all science students, and in science and engineering, there's something called as a root cause analysis. That means you have a problem, you have a challenge in front of you, and if you want to tackle the challenge, if you want to solve that problem, the first thing that you need to do is you need to identify the cause of the stress, right? So that is what you have to do here also. You have to identify what are the causes of the stress. It could be anything, it could be different for different people. It could be that you have very less time for the exam and you're not prepared thoroughly. It could be that you have prepared but the workload is still very heavy and you're feeling quite overburdened, you're feeling overwhelmed. Then there could be other problems in other areas of your life which have nothing to do with your studies, which have nothing to do with the exam. It could be some family problems, it could be some other friend problems, but problems in other areas of your life that trickle down onto your work, they trickle down onto your studies. Then, as Tanushree ma'am rightly said, yeah, on this day and age you have a lot of social media distractions, right? So that is one thing which causes many people to procrastinate, and then that's one of the reasons that ultimately leads to stress. Then insufficient sleep or insufficient relaxation is also a cause of stress. You may find that you're fighting this battle quite alone. You may feel that there is nobody to talk to, that you may feel quite alone here. So there is a lack of support maybe from your family, peers, friends, colleagues, mentors, teachers, etc. So that could be a cause of stress. Or it could be simply lack of confidence. Maybe you have prepared, maybe you have put in the hours, you have worked hard for this, but still just a simple lack of confidence is what is causing the stress in you, and because of which you think that you may mess up the exam, right? So there could be other reasons also. So the first thing that you have to do is you have to identify what exactly is the cause of your stress. Once that is done, that's half the battle won. Because once you know what is causing the stress, there are different ways to deal with the stress. For example, eating healthy. Now, this stress need not be related to just your study or just your exam. It could be applicable to any other aspect of your life, and these are some generic ways by which you can deal with stress. The first one is eating healthy. You're all life science students, I don't need to tell you that healthy eating is directly connected to a healthy mind and a healthy body. If your mind is healthy, if your body is healthy, then you can learn, you can absorb, you can contain the information, and you can reproduce it during your exam. So eating healthy is very important. Then exercising, as again Tanushree ma'am rightly said, you need to exercise. Give at least 10 to 15 minutes per day for exercising, because that burst of exercise will release a burst of endorphins in your body, which is like a feel good neurotransmitter from your brain. And that feel good neurotransmitters just simply helps you absorb information. So exercising is very important. Time management is already covered. Like I said, if you feel like you're battling this alone, and it's a huge task in front of you, it's better to talk to someone. It could be anybody, it could be your family, it could be your friends, extended family, your teachers. We at Biotechnica, we are always here, we are always ready to talk to you, even if it's not related to your subject. So talk to anybody. It could be your cat, it could be your dog, but you have to talk to someone to release out the things that are holding inside you, the things that you think are causing stress for you. Then you have to create a comfortable environment in which you will study. Now, comfortable environment, it could be anywhere. It could be in your home, a nice little cozy study table where you have things that you like, your diaries, your books, your laptop, everything in front of you. It could be your college library, it could be your community library, it could be any space which you can create a comfortable environment in. You can sit there and you can study, right? And here again, like it was mentioned earlier, it's important that you keep your phone switched off or silent and keep it inside some cupboard so you don't have access to it, or make sure that no notifications are disturbing you. And lastly, practicing mindfulness. Now, what is mindfulness? Mindfulness is a state of awareness. You're very much aware of the present situation. This awareness transcends a lot of boundaries. Okay, it's not like you're becoming a big yogi or something, but this mindfulness is being very much aware of the present, the current present situation, your emotions, your feelings of your body. That is mindfulness. You have to accept whatever you're feeling right now. Okay, are you feeling happy, are you feeling sad, are you feeling butterflies in your stomach, are you feeling your heartbeat increasing or your heart pumping a lot of blood? So that acceptance has to be non-judgmental. Okay, if you're feeling pits in your stomach, you shouldn't become sad. Oh no, I'm feeling stressed. No, you have to look at it from a third person point of view. So a non-judgmental acceptance of the present state of your mind and your body is called mindfulness. Now, I know I said a lot of big words, and it's easier said than done, but there are some tips and tricks that you can use to practice mindfulness. And the first one is focused breathing, with the emphasis on focused breathing. So focused means your entire focus should be on the breath. For example, 7 seconds breathe in, 7 seconds hold, 7 seconds breathe out. That's one of the examples. There are different types or different ways in which you can do. So if you do yoga, if you do meditation, you are actually following mindfulness. So focused breathing is when you're completely focused on your breath and nothing else. You are not distracted by any other situation, any other thing. If your mind wanders, just quickly you have to get it back to your breath. That is focused breathing. Meditation is a tool for mindfulness. Meditation is not equal to mindfulness. So again, if you are doing continuously yoga, towards the end, your guide or your teacher would take you through this guided meditation. So that is one of the ways by which to practice mindfulness. Even the fourth point, body scan. Body scan is when they take your attention to every part of the body to see whether it is paining, to see whether there is some stress situation over there, whether you think it's fine or not. Okay, so like for example, focusing on your forehead, focusing on your ears, focusing on your shoulders. So this is a body scan analysis, which is also done during guided meditation. Mindful eating is when you sit with the food on the table, no distractions, no phone, no TV, no nothing, no talking, just focus on the food that you're eating. That is mindful eating. Now, other activities like music, dance, walking, painting, or even journaling for that matter, just take out some time for it. Maybe not every day, but maybe once a couple of days you can do that, because these are different forms of mindfulness. They bring your attention to that particular activity that you're doing, and it keeps your attention to that particular activity. So your focus is not going here and there, your focus is on the activity that you're doing. And lastly, gratitude. Every day be thankful for everything that you have today. And if you're thankful for today, be sure or be hopeful that this will continue for the coming days. So with this, I'm not going to take much of your time. Remember, your future self is going to thank you for not giving up today. All right. So thank you so much. I'll pass on the controls back to Shekhar sir. Thank you.
All right, welcome back. I'm sure you had an amazing webinar today. I know we have less time now. We've already overshot the time. So now is the last part of the webinar where we are going to give you a feedback form. Please fill it so that we know how did we fare, how was our performance during the webinar, whether we were able to help you or not. And also let us know what are the problems you are facing in your preparation so that we can guide you. Now, after that, after filling this form, you will get an email which will have your 100 days, 100 topics ebook PDF where you will have complete elaborate list which you should study first, prioritize first, solve questions more from that topic, and then you're going to qualify. So all the best to all of you and have a great evening. Good night.